Hello my dear students. So today I have brought one amazing video and you will be thankful later that I have brought this video for you. This video is the video for the diagrams. In this we are going to discuss all the important diagrams or the possible expected diagrams for 10 ICSE board exams. Exam is so near and we are so worried how to prepare well. So because we can't skip the diagrams in biology, this is the one of very important and life-changing video for you. So do not skip the video, watch till the end and please sit with pen and paper so that you can also draw along with me, and you need not to draw it again and again. Just once if you'll draw with me, it will be very helpful, and until the exam you don't have to worry about the diagram. So without any further ado, let's start our class. Okay students, so without any delay we are going to start our class. So here I have listed all those very important diagrams which are like going to come in your exam, or maybe important. Not every diagram will come, but all of them are important. You don't know which of them are going to come. So basically all the diagrams we are going to do. So in one video, or maybe I'll make the part of it. So one or two video, everything you will be see, and that means once you have to revise and you will be very ready, completely ready for the exam. So okay, so what are the list here? Let's see the list first. So first is the phases of karyokinesis. Here 4 phases are there. P-mat, we call it P-mat, like prophase, metaphase, anaphase and telophase. All the 4 phases diagram we are going to see. Nucleotide, that is the one of the very important diagram. Root hair cells, in this root hair cells we will see as well. The plasmolysis, when the cell gets plasmolized, how is it shaped that time, that also we are going to see. Stomatal apparatus, very important. Chloroplast, and these are very easy diagram, you'll see how easily we can draw it. Okay, chloroplast. TS of artery or vein, TS means transverse section. Transverse section means this section of any artery or vein, how is it going to be drawn. Then there will be human excretory system. Human excretory system, we are going to see all the things like kidneys, ureter, urinary bladder, everything, and LS of kidney. See, this is two different things, because here we are going to see the longitudinal section of kidney. Like if this is one kidney, will give a cut in between, and this part, left side part, we are going to see from this side, and how does it look from this point of view, that we are going to draw here. Okay, then nephron, one of the very important part, like a key unit of kidneys, so we are going to see that also. Neuron, related to your brain, and so we'll see the brain also. Brain's diagram also will be important here. Reflex action or reflex arc. Okay, now both is the same thing that we are going to see. LS of eye and correction of its defects. So we all know eye has so many defects, that correction of that, like which lenses to be used, so that to correct it. So all the diagrams, and obviously the eyes diagram, very important. Then LS of inner ear, this is also one of the very important from the sense organ. And these are the important diagrams. So LS of inner ear, how does ear work, and how the like sound passes, what are the parts, this we are going to discuss here. And at the last, sperm. Also two things I have not mentioned, but I'll be like explaining it while drawing all the diagrams. That is male reproductive organ and the female reproductive organ. That also will be one of the very important part. So these are the total list which we are going to discuss. Today we will not be discussing all of them, just few of them. Like obviously I don't want to make you feel burdened, because there are list of at least 15 to 16 diagrams, and in one go if I draw all of them, then you will be very confused. So what are we going to do? We'll part wise we are going to discuss it. So maybe I'll take 5 in one, or maybe in one video I'll take 5, and then according to the time, I just don't want this video to be too long, so that you don't feel burdened. Like it will be a chill moment for you. Once you have done revising all the things, just look at this video, draw some of them like along with me, and then you'll be ready for that. And that will give you short marks, means each of the diagram comes for 3 marks, 4 marks, 2 marks, 5 marks even. And obviously there must be some questions attached to it, like reasonings attached to it. So I don't want you to like lose this easy mark. So just look at this, it should be refreshing. So let's start. We don't have to waste our time. Let's start. So let's see how many videos we are means how many diagrams we are going to make. So for that, first of all, we'll start with phases of karyokinesis. Okay, phases of karyokinesis. Phases of karyokinesis. Okay, see, in phases of karyokinesis we have 4 phases. I told you that first is the pro phase, so that we are going to draw first. So in pro phase, we know we have to make two cells. One is animal cell and one is the plant cell. Okay, so in plant cell we know there are two membranes. One is the cell wall, one is the cell membrane, and this is here it has only cell membrane. Okay, after that we will have will make the disappearing cellular membrane. Disappearing, it is disappearing nuclear membrane. Here also disappearing nuclear membrane, and then we have disappearing nucleus. Disappearing nucleus. Okay, so this much is here, and then we'll draw like this is the centriole, and there is the ester, and this is only in animal cell. Okay, and then there is spindle fiber. Okay, same thing here, and this is the ester, and this is the spindle fiber. Okay, see, here we are making for us total number of chromosome is 2. It is the easiest one, that is why we are drawing that. Whatever is given, like let us suppose it is given 3 or 4, then you need to draw that. Okay, so for me it is 2, that is why I am drawing only 2 chromosomes. So chromosomes will be like pair of chromosomes. Here will be drawn 2, here also 2 chromosome. Okay, then this much is there, and let's label it now. First of all, this part is the separate part because it is only present in animal cell. This is ester, and this is centriole. Okay, this is centriole. Fine, and then okay, this is the common part. Oh, I haven't drawn it yet. Let's draw it here. Here only, see, here centrioles are not there, only spindle fibers. Only spindle fibers, no ester, nothing. Okay, let's see. So this is the spindle fibers. Okay, so this is nuclear membrane, and then there is, this is although nucleus, but no need to label it. Okay, this is not that important. Then we have, okay, this one. Then we have this, which is chromosomes. Okay, so this is the pro phase. Fine, now let's do the next phase. Metaphase. Second is metaphase, and again as we have taken 2n is equal to 2. So this is what we are going to follow in all. Okay, so two types of cells we are going to draw like earlier. One is the animal cell, one is the plant cell. Okay, so plant cell will have two membranes, one is cell wall and cell membrane. Okay, and then here the chromosomes we know along with equator. Chromosomes are here along with equator. So we have two chromosomes along with equator, okay, in a single line. Along with equator, this is done. And then we have this obviously centriole and then ester only in animal cell, okay. And then from here the spindle is this way connected, okay, ester, centriole, and this is this way connected. And the same thing here, spindle will be connected. Obviously it will not have any centriole or ester, these things will not be there, rest everything is same. And again here what you need to do, you need to write, you need to mention chromosomes along with equator. This you need to write, okay. Rest everything will be the same, labeling will be the same, like this is ester, okay, this is ester, this is a centriole, okay, this is both for them spindle fibers, okay, this is same, everything will be the same, right? So second phase is also done. Now let's do the third phase. We'll make the third phase, and third phase is anaphase. Okay, third phase is anaphase. So for anaphase again we are going to draw two cells. One is the animal cell, one is the plant cell. Okay, so here what we will draw now, this chromatid will be drawn along with opposite poles, right? Along with opposite poles. Instead of chromosomes, now this is chromatids, okay, along with opposite poles. And then this will be like this way, okay. And again this is centriole, ester, okay, ester, here also centriole and the ester, here only spindle fibers. So this is this way. And now you see this here labeling will be done. So let's label it. Okay, let's take this one. So label will be the same. This is the same, spindle fiber, spindle fibers, okay. And now instead of this chromosomes, this is chromatids. This is the only thing. Again this is ester, this you need to draw, and then centriole. So I already have told this thing, so you can easily do it, okay. So you know what, here the questions can be like, maybe one phase will be given and the questions will be given from one phase, and they will ask draw the previous phase or draw the next phase. This way the things can be done, okay. So for that case we need to understand that we have to be ready for any of the phase. Maybe one phase is given whose question answers, the question answer from that phase is been asked, but next they can ask you draw the next phase. So you have to be ready, and you must know which is like the phase which are coming after, okay. Like anaphase is there, what will come before it, what will come after it, that should be completely clear in your mind, okay. Let's understand the next phase, the fourth phase. And the fourth phase is telophase, okay. Telophase, okay. Now in this phase again the two diagrams, but here In animal cell there will be the furrow formation, and in plant cell there will be only one plate formation. Okay, then again the chromatin will be there, nuclear membrane will be there, and then that chromatids will be decondensed. Okay, again the same way here also. Okay, this will be there, and now one nucleus will also be appear. Nucleus will also appear. Okay, so this is done. Okay, so this way this will be made, and then this is done. So first of all, this is the nuclear membrane. Okay, labeling is very important, keep it in your mind. Then this one is condensed chromatin fibers. Okay, condensed chromatin fibers. There is nucleus. Okay, here in animal cell, furrow formation, and in this you see this one, this is the cell plate formation. Okay, so obviously this is animal cell and this is the plant cell. I hope it is very clear. So this is how it looks like. Okay, this is how it looks like. So this was the 4 phases which we have drawn clearly. Now let's go to the next shape, next diagram. Okay, so next diagram is of this much is done. Okay, so we are going to do the next diagram. See, we have done 4 of the phases, and all the 4 phases labelings are very important, and I told you how which type of questions also will be there. Now next is our nucleotide. See, nucleotide is very important diagram, and what you need to understand here. So next is nucleotide. Okay, in this diagram we have to understand 3 things. One is the pentose sugar. Okay, second is the nitrogen base, and third is the phosphate group. These are the 3 things you have to keep it in your mind while drawing the nucleotide. Now let's draw it. So first of all, we have this pentose sucrose. So this pentose is 5. This way 5 sugar should be there. See, then in the top, and that is on the apex, there will be no carbon, there will be oxygen, right? Then 1 carbon will be here, 2nd carbon here, 3rd is here, 4th is here, and the 5th carbon. The 5th carbon has to be drawn out, so we have to draw it this way, and here is the 5th carbon. Okay, now in the first one, here will be the nitrogen base. This will be attached. Okay, nitrogen base, we need to understand the type of the nitrogen base. Nitrogen base type is AGTC. Okay, adenine. Okay, just give me 1 second. Let me write it clearly. Adenine is making double bond with thymine. And then there is second one is guanine making a triple bond with cytosine. This is all the nitrogen base, types of nitrogen base. Adenine double bond making with thymine. Guanine is making triple bond with cytosine. This you need to understand, you need to learn, and if you want to learn it, just learn it by AGTC. Okay, AGTC. This much you need to learn. Okay, then there is this phosphate. Here we will write the phosphate. Okay, you need to write this phosphate group. You need to write this. So understood this? See, all the diagrams has to be drawn very neat, very clean, and drawn with the pencil. Of course, there must be written the title on the top, and then the drawing should be done. All the labelings are very, very, very important, and this labelings has to be done. Here you need to write pentose sugar. Okay, that you need to write. Everything is done, and this diagram, see how simple, very, very easy diagram this is done. So this was the very easy diagram of nucleotide. Now we'll draw the next one. Okay, so next diagram is of root hair. See, this is also one of the very important diagram. So for this root hair, we are going to draw in this way. So first of all, so this is a double membrane thing, so means obviously it will have a cell wall and the cell membrane. Okay, it will have the vacuole like this. Okay, one nucleus will be there, and then rest part will be chromosomes filled in it. Okay, now let's label it. This is done. Okay, just we need to label it. Labeling is also very here. This is the cell wall. This is the cell wall. Then this is cell membrane. This is cell membrane. Okay, this is a root hair. This is root hair. Okay, this is the large, you can see this very large size vacuole. This is nucleus. Okay, and then this is chloroplast. Okay, this is chloroplast. Everything is done. So this is the whole root hair diagram. This is very simple. You can see this is very, very, very simple. But one stage is also been asked many times, and that is whenever if there is if you have to show the plasmolized, okay, if you have to show the plasmolized cell, plasmolized root hair, then how will you write? So let's plasmolized. If there is plasmolized cell, then how will it be drawn? See, the first part, which is cell wall. Cell wall is a dead part, so it will not be affected anymore with this. Okay, cell wall is the dead part, right? So this is how it will be drawn. Okay, so this is how it will be drawn. Now cell wall is the dead part, so it will be here. Now we need to draw the cell membrane. Cell membrane will be shrinked. Okay, so cell membrane will be shrinked so that it will come this way, and very, very less. See, earlier it was still here, but now it is here only. And then the vacuoles will be very smaller in the size. See, the vacuole were very large sized, but now it is very small. And then there will be a nucleus, very, very tiny. Okay, then there will be a nucleus. This is now what is there here? What is there here? Here is that hypertonic solution. Hypertonic solution, right? This is the hypertonic solution filled in the whole empty places, okay? That like places will be occupied by the hyper, and that is why this cell membrane is just dying, okay? It's just getting dying, okay? Now let's label it. Labeling is very important. Here this is just the cell wall. It will not be affected because it is a dead part, okay? Then this is the hypertonic solution. I already have written this. This is obviously the root hair. This is already the root hair, and then this is the cell membrane, okay? This is the cell membrane in a corner. In a corner it has come. This is the vacuole, and now this time we are writing small size. A small sized vacuole. Vacuole. This is the nucleus. Okay, this is the nucleus. So this is the whole part. This is the way. So many times asked questions is been asked that root hair, draw a root hair and then plasmolized cell. Means when it will be plasmolized, and how does it look like? So it looks like this. I hope it is very clear and very important diagram and easy one. Okay, now let's draw the next. Root hair has been done. Now we are going to draw a small stomatal apparatus. Okay, stomatal apparatus. Okay, stomatal apparatus, very important. Okay, here you don't have to draw anything, like just two kidney bean like structure. Okay, and then again like this. Okay, two kidney bean structure, and then there will be the epidermal cell. So it will be drawn in this way. Okay, I hope it is clear. Now it will have the vacuole of big size, and then it will have the nucleus. Let's draw it from, okay, I hope it is visible. Okay, this is nucleus, and then again the chloroplast. This is filled with this. Now let's draw the let's do the labeling, and labeling is also very easy. This is what? This is the epidermal cell. Okay, this pore is called stoma or stomatal pore. Okay, stomatal pore. I'm just writing it, stomatal pore. Somewhere written this and somewhere stoma, you can say stoma. This is guard cell. Okay, this is vacuole in the guard cell. This is the nucleus. Okay, and this is the chloroplast. See, it depends. Chloroplast. You need to label as much as possible according to the marks given for you. Let us suppose if this is a 10 marks question, at least 10 marks question, not 5 marks question, at least 10 labeling should be there. If this is 3 marks question, obviously it is a 3 marks question. Okay, it will come in 3 marks. So you need to label at least 6. So how many levels we have done? 1 2 3 4 5 6 levels we have done. So this is a 3 mark question. So you must have to do this, and then only you will get the complete marks. Okay, I hope it is very clear. Stomatal apparatus is one of the very easy diagram. Okay, let's move to the next. Next is our chloroplast. Okay, next diagram is chloroplast. Chloroplast. Okay, chloroplast. You need to understand the structure first. Chloroplast is a double membrane. Chloroplast is a double membrane figure. So one membrane, and then let's draw one membrane outside, So second membrane. Okay, so outer and inner membrane is done. Now we will have the coin like projection making a stack. Okay, this way one more. You can draw as much as you want. Okay, you can draw one more like this way. Okay, so this is one. And then we have attachments, frets, which is called frets. Okay, this way attachment, and it will have a DNA also. Okay, it will have DNA, and then inside it there is chloroplast, chlorophyll. Inside it there is chlorophyll. You can see this chlorophyll here also, chlorophyll. Okay, and then it will have all way ribosomes, this way ribosomes. Okay, now let's label it. So first of all, this is the outer membrane, then this comes the inner membrane, because this is a two membrane figure, double membranous. Okay, so this is called this stack, complete stack is called the, see, first of all, one of them is called thylakoids. Thylakoids, and then this platform is called stroma. See, earlier we have done stoma, and this is stroma. Okay, this yellow part which you can see is ribosomes. Okay, this is your DNA. Okay, this is thylakoids, and this is the complete one, like this one. This complete one is called grana. I hope it is very clear to you. Okay, I haven't missed anything here. Yeah, if you want to label this, you can label it also chlorophyll. Again, as I told you, according to the marks you need to do the labelings. Okay, however marks is there, just double the labelings. Okay, so that will be done. You must see, this is the double membranous figure. One is the outer membrane, second is inner membrane. Thylakoid is the one sac. Okay, one coin like structure is called the thylakoid, and if you'll make the stack of it, if you'll pile up it, then that will make the grana. Okay, this platform is called stroma, and in the platform, this yellow kind of things you can see, that is the ribosomes, and it has a DNA as well, chromosome as well. And then inside the like thylakoid, inside it there are so many chlorophylls available. Okay, then exactly here the photosynthesis takes place. Okay, exactly here. Transverse section of artery and transverse section of vein. So we'll draw it in one go only, so that it will be easy for you to understand as well. So first of all, what are we going to draw? See, it is a 3-layer thing, so we'll draw 3 layer. So you see, this is the lumen. This lumen is so narrow. See, artery's lumen is narrow and veins lumen is broad. So accordingly we'll draw. And the second layer is more fibrous. So you see, there is a gap, means this is thick enough. Okay, then the third layer. Okay, now in this, what will we do? We'll draw the first, that is the lumen. Lumen should be the wider. Okay, it is narrower, it is wider. I'm making it more narrower so that it will be create the more difference. Okay, this is the way, not this much, but yeah. Okay, this is fine. Now this is the narrower one. This is the narrow one, this is the wider one, and the second layer is not that fibrous like this. Okay, its second layer is more fibrous, so it is not that like that. in this way. Okay, it has cells. Okay, it will have a cell structure like this. Okay, it will also have the cell structure like this. Okay, so this is how we are going to draw this. Now let's okay, give little bit differences here so that it will look slightly different from the other layer. The same thing we are going to do here. Okay, I hope this is very clear. Now let's label it. While labeling, we need to understand this. This is the outer one. Okay, this is called tubica. Tubica external. External. Okay, tubica external. Tubica is the layer. Tubica means layer, and external means outer layer. And then comes the middle layer, and this is called tubica. Tubica media. Tubica media means the middle layer. Okay, now when you write tubica external, you also have to mention how is it made, like what is it made from. So it is made of fibrous connective tissue. Connective tissues. Okay, it is made of fibrous connective tissues. So you have to be very peculiar about writing the what is it made of. Okay, then only you'll get the full marks, otherwise you'll not get the complete marks. Then tubica media, it is made up of smooth muscles. Okay, it is made up of smooth muscles. Okay, this is made of smooth muscles. This is done. And now the last layer, third layer, this layer is called endothelium. Endothelium, you must have heard epithelium. Epithelium means epi means outer. Okay, epithelium, outer layer, endothelium, inner layer. So this is, you see, this is the inner layer. Now this cavity, and this cavity is lumen. Lumen. You see, all the labelings is done. I hope it is very clear to you. What you need to understand is this: when you see the thickness here and you see the thickness over here. So this thickness is very less and this is very thick, and that defines this is artery and this is vein. Also the lumen size. Okay, lumen in artery is narrower and in vein it is wider. So these are the basic differences. If you know that, if it is clear in your mind, you can easily draw the TS of vein and TS of artery. This is very easy. Okay, I hope this is very easy. Now let's understand the excretory system. Human excretory okay, human excretory system that we are going to draw now. Okay, this is also very easy. Here first we are going to draw the kidneys. Okay, kidneys are like when we are drawing, so this is the right kidney, this will be the right kidney, and this will be the, although this is left side of mine and that is the right side, but when the drawing will be done, this will be called the right side and this is the left side. This is all for heart, also for any diagram it will be applicable. So first we'll draw the kidney. Okay, so one kidney, and this cap is also known as, you know that cap, let me make it more clear. You know that cap, this cap is called adrenal gland. Okay, and then there will be one slightly, you see, slightly up I am going to draw the another kidney. Okay, this is slightly up, it will also have the adrenal, but little just this is little this side, so I'm just going to draw it a little straight. Okay, now it will have an adrenal gland like this. Okay, now we have very important thing to draw, and that is the vein and artery. So first of all, this is the aorta. Okay, here see, when you'll draw, I'm drawing it with red and blue. I can use that colors because I have it, when you have to draw this, you'll draw with the pencil, so you will not have the option to draw it with different colors. Okay, so what are you going to do? You are going to place some arrows. Okay, that will be, you are going to place some arrows, like it is coming inside, it is this side getting in, so this is getting inside. Okay, so this is the arrow is very important, because you don't have the option to you don't have the option to choose the colors. Okay, you can't have the colors here, option of colors will not be there. So next is the inferior vena cava, and next, so this is where this will be here, somehow like this, and then okay, this way, somehow like this. Okay, here the arrow is outer. Okay, arrow will be outer, here is also arrow is outer. In this way, this has been done, this has been done in this way. I hope it is clear. Now we are going to draw the ureter. Okay, this way, and then this has the sphincter muscle. Okay, I hope it is clear. Now let's label it. So this is the left kidney, this is the right kidney. Left kidney, right kidney, this is aorta. Okay, this is inferior vena cava. Okay, this one is done. Now this is adrenal gland, we need not to mention it. Okay, this is ureter, this is urinary bladder, where the urine gets collected. Okay, then this is urethra, this is the opening. Okay, urethra, and this one, red one, is sphincter muscle. Okay, sphincter muscle, and this much is done. And okay, here the important part, and that is this one, this layer. You see this layer, I'm just doing this orange so that you can see the highlighted part, like we are just interested in the arrow. When this arrow is there, so what will you do? Write renal artery, and then this arrow, this outside arrow is there, so you'll write renal vein. Okay, see, renal word is related to your kidneys. Okay, whenever you use kidney, that is renal. When you talk about the lungs, then you'll use pulmonary. Okay, these things I think you know all these things. So this is the all about human excretory system. How does it behave? How does it work? This you already know, and in the separate video I'll discuss those also. So this is the whole part. I hope this is very easy, and like one of the very easy and important part. And the questions will be like role of renal artery, role of renal vein, how does it work, which goes away from the heart, which goes to the heart. This way the questions will be asked and the drawing will be the same. So let's draw the next. So here we have done the complete system, and now one by one we'll see the kidney. Kidney is also one of the very important part. So LS of kidney. LS means the longitudinal side. Longitudinal side means if something is like this, we'll cut in long along with, and then those two parts will get one part. This side we are going to see what is inside. Okay, so that is the longitudinal kidney. So let's draw first the kidney. Okay, this is somehow like kidney, and in this there will be pyramid. Okay, Okay, and this pyramid kidney is opening on the pelvis. Okay, see here, one more layer has to be drawn. This is the 2 layers here. Okay, so now this will go in this way, pelvis, and then this will come after like this. Okay, it will come after like this. It will have the striped appearance, and then okay, this is how it looks like. Now just label it. So first is this one. The first is renal capsule. Okay, the second part is, this is renal cortex. Okay, renal cortex, and then inside part is renal medulla. Now this is medullary pyramid. Pyramids. Okay, this is the opening. See the pyramids opening. This is called renal pelvis. Okay, this is the ureter, and then here comes the 2 vein and artery. One which will go inside, one which will go outside. So inside one is called renal artery, and this outside one called renal vein. I hope it is very clear. Very easy and simple diagram. You see this, very easy and simple diagram. Just one kidney, inside it there are some medullary pyramids drawn, and the pyramids will show the striped look, and then inside it there is this is the ribosomes, this is the structures in this way, and then there will be the capsule, cortex, medulla, and medullary pyramids, everything. And this labeling is done, drawing is done, and I hope this is very easy for you. Okay students, we are done with the important diagrams part 1. So in this part we have discussed almost 9 diagrams, which were all very important and simple one. So I am pretty sure you must have been drawing with me, and was amazing experience for you all. So if you have any doubt left, just do let us know. Not only for this topic, but any topic of biology or any subject, just do let us know in the comment section. We'll try to solve this out as soon as possible. Again, if you like this video, just hit the like button, share it with your friends, and please subscribe to our YouTube channel Mida Guru for more such contents. Also, our Instagram contents will amaze you, so just follow us there also. So let's meet with next part. In this, that part we are going to discuss more diagrams. So let's meet then. So till then, keep practicing and keep learning. Bye bye.